Proteins that regulate DNA transcription recognize specific DNA sequences through discrete DNA binding domains. These domains are, in general, relatively small, less than 100 amino acid residues. Many prokaryotic DNA binding domains contain a helix-turn-helix motif. In the following two chapters, we discuss wider range of DNA binding motifs in eukaryotic DNA binding proteins, such as zinc finger, leucine zipper, helix loop helix, helix-turn-helix, and something like that. In this chapter, we'll focus our discussion on the two bacteriophage proteins, since they are amongst the best understood DNA regulatory proteins. This slide explains life cycle of phage lambda. This phage lambda undergo two different life cycles, lytic and lysogenic cycle. After adsorption on the host cell, such as E. coli, they just inject. It's a linear form of DNA. Inside cell, this linear form is converted into circular type, and then it could go either lysogenic or lytic phase. So basically, lysogenic phase. This DNA, phage DNA, just incorporated into E. coli chromosome at specific site and behave like a chromosome, E. coli chromosomes. So whenever E. coli divide, just phage DNA also replicate and inherit it into the daughter E. coli's. Instead, lytic phase just replicate its own DNA by rolling circle mode, and in addition, viral coat proteins and translations occur, and later the assembly producing the daughter phage, they just escape out of E. coli. So the initial the gene regulations determining the phase of this phage newly introduced DNAs, either lysogenic or lytic. There are three different transcripts initiating from PL and PR. The early transcript going two directions, and middle transcript, the following the early transcript, start from the same position and going further, both directions. And late transcript initiate from different promoters. The early two transcripts are responsible for determination of the life cycles, either lysogenic or lytic. This is just showing the regions responsible for the early transcript. So there are two important structural genes, repressor at the left side and crow on the right side. In between, the expanded version indicating 3 operators, OR1, 2, 3. So basically, OR1, OR2 is overlapping with the crop promoter, while OR2 and 3 overlaps with repressor promoter. You can see the concept of promoter and operators. Three of operators are aligned, showing certain features. They are highly conserved in green colors, and also each one shows palindromic symmetry, twofold symmetry. So according to the axis at the position 9, when you rotate by half turn, they're going to produce similar sequences. The significance of this palindromic symmetry in operator is that the proteins binds to this region must also have a twofold symmetry. So anything binds to here, when it rotates by half turn 180 degree, the region also must bind this one. You may see the structures based on this one soon. This slide explains the situation in lysogenic bacteria in A, repressors, and RNA polymerase binds to the switch region in the lysogenic strain of E. coli. The repressor binds to OR1 and OR2, thereby turning off synthesis of crogene. The repressor also works as an activator for its own promoter by facilitating RNA polymerase binding to the repressor promoter through its binding to OR2. In B, explains lytic cycle. The crop protein binds to OR3, blocking the RNA polymerase from binding to promoter for the repressor. So only CRO genes just turning on the right genes that are responsible for the lytic cycle. So two proteins, repressor, red one, and protein in green. So there is a topic in this chapter, how these two proteins binds to these regions by base sequence specific mode. CRO protein from phage lambda showed this kind of structures having helix-turn-helix. How do repressor and CRO recognize the specific operator regions and achieve this subtle differential binding to the switch regions? First glimpse into the structural details of this system was obtained in 1981 when the group of Brian Matthews at Eugene, Oregon determined the crystal structures of protein. We are looking at protein from phage lambda, small size, 66 residues. In A, alpha carbon, but only using alpha carbon. In B, the schematic diagram of subunit structure. 3 alpha helices and with 3 beta pretty sheet. The helix 2 and 3 forms helix-turn-helix motif responsible for the DNA binding. Proteins, they form dimers, and actually dimer is made by interaction between beta strand 3 from each subunit, 3 and 3. Another important point is that the helix-turn-helix loop in this dimeric structure position apart by 34 angstrom, which corresponds to the 1 ton of DNA, 10 base pairs. This source strongly suggests these regions are responsible for DNA binding. Lambda repressor is much larger, 236 amino acid, and is composed of 2 domains, N and C. 2 domains could be separable by treatment of Fourier's, so only N-terminal domain was determined for each structure, showing here. Mainly by composed of alpha helices, but the 2 regions in the blue and red similar to the crow helix-turn-helix. C-terminal is responsible for making dimers. However, N-terminal domain alone could make dimers and also be functional. Since these protein structures were determined in the absence of DNA, there was at the time no experimental evidence that these alpha helices were involved in DNA binding. But the indirect evidence that these red helices in these dimers just position apart by 34 angstrom strongly suggests that these red helices may be involved in DNA bindings. Given that the operator that this protein binds to also shows two-fold symmetry. So specifically they have some certain orientation, not 20 degree. Based on such observation, Matthews just suggests this kind of binding mode, where red one being inserted into major group of operator.